human activities increase the release of atmospheric CO2, leading to global changes such as global warming or ocean acidification. Consequently, the amount of CO2 dissolved in the ocean increases and disrupts the carbon equilibrium, causing the reduction of seawater pH. It has been observed that this phenomenon may have harmful effects on species behavior, skeleton calcification, or physiology. All the oceans are concerned, and the Mediterranean is not an exception. An increase of 4 degrees of temperature and a decrease of 0.4 units of pH has been predicted for the year 2100. Therefore, it is one of the most sensitive seas to climate change. Hi, I'm Keyla Plichon, a first-year student of the MARES program and an intern at the LOV. I've been working on a project that aims to assess the impact of ocean acidification and ocean warming onto Mediterranean corals. This project is part of the Shock STEAM research and the 4 Oceans Mobile program to assess the effects of global change in biochemical cycles and marine organisms. More precisely, it is part of the PhD thesis of Chloé Carbon, supervised by Nuria Teixido, Steve Como, and Jean-Pierre Gattuso, with the help of Samir Aliwan. Ocean acidification may have negative consequences at different scales of corals, for instance, reproduction. Since reproduction is essential for species conservation, the project aims to assess the effects on the gametes production and the early life stages of 2 endemic Mediterranean corals, Astralis calicularis and Cladocara caspitosa. Samples have been collected on the island of Ischia, where volcanic pure CO2 bubbles locally acidify the environment, approaching the conditions predicted by the IPCC for the year 2100 under one of the worst scenarios. We compare coral colonies living in these low pH conditions in pink with colonies living in sites where the pH is ambient in blue, around 8.05. Reproduction is monitored by the analysis of the gametes through histological cuts and the energy dedicated to reproduction, thanks to lipid and symbiont quantification. The results of these experiments are expected for August, so stay tuned. Finally, the early life stages of Astralis calicularis' life cycle are also studied by exposing larvae to different combinations of temperature and pH to estimate their recruitment success. I followed their development by regularly reporting if they were in a planula or polyp stage, if they were settled or free, their mortality, size, and skeleton density. The first conclusion of this experiment is that juveniles have more difficulty settling under acidification. Indeed, the polyp base was larger in the ambient pH conditions, while in the low pH conditions, the base was minor and polyps could detach from the substrate. We can therefore say that future climate conditions will decrease the recruitment of Astralis calicularis. A new larvae experiment will start this summer to assess their possible adaptation to ocean acidification. So the next student will have to maintain the tank's parameters, keep the larvae alive, and continue the gametes experiments with Astralis calicularis. I encourage the new students to apply to this immersion project. You will have the opportunity to contribute to experimental design and submit new ideas as I did this year. You will have the chance to work in a beautiful lab, meet very interesting people, and discover these colorful corals as well as other Mediterranean organisms.